All right, we're here for information about different E. coli strain genotypes and how to actually understand all that info. The information listed in a strain's genotype tells us a lot about the cell, what it's equipped with, and what it's capable of doing. And all of this information helps you decide whether it's going to work for your experiment or not. So in this video, we're going into the different properties that a strain's genotype tells us about and how to make sense of all of it. Just before we start, I do want to say that this is not an exhaustive list, but it does cover a lot of ground to get you started. Also, for this video, we're just focusing on E. coli E. coli cells, and I'm not going to talk much about the different strains, but I will mention examples of specific strains to hopefully just make it make more sense. Let's start out with what a genotype is, and then we'll dive in deeper from there. When it comes to competent cells, and E. coli in particular, the genotype contains information for the gene's mutation types, allele numbers, antibiotic resistance, insertions, fusions, inversions, and deletions. The genotype for E. coli cells only includes genes that have a loss of function mutation. For example, in GoldBio's GB5 alpha E. coli cloning strain, the genotype contains REC-A1. This notes a loss of function mutation for the REC-A1 gene. Something to notice about how the genes appear in a genotype is when looking at the gene name listed in the genotype, they're listed as a three-letter combination in lowercase italics. There's a little more to it because if we look back at that REC-A1 mutation, you'll notice that there are 4 letters involved and a number. Different genes affecting the same function or pathway are identified with a different uppercase italic letter, so you'll notice the A in the REC-A is capitalized. And in the case of different mutations in the same gene or different alleles, a number is added to indicate the specific mutation. If you happen to see a superscripted lowercase q, it indicates a constitutive mutation. This means continuous unregulated gene production. So lac-I with the superscripted q would be a mutation that results in constitutive expression of the LAC repressor and inhibition of the LAC promoter. The genotype also shows us information about insertions, deletions, fusions, inversions, prophages, restriction-modification systems, and even phenotypic traits like antibiotic resistance and sensitivity. So let's take a moment to look at each of these and how they would be noted, starting with insertions. Insertion of a transposon into a known gene is indicated as a gene and then 2 colons and then the transposable element. And just to clarify, transposable elements have a mechanism of their own to move and insert themselves into a host organism's DNA. So let's look at an example of how this would appear in a genotype. Cis C95 TN10 indicates the transposon TN10's insertion within the cis-C gene. And just to remind you, the 95 in the cis-C95 is the mutant allele number 95. TN10 and TN5 are both common transposons. TN10 is a transposon carrying tetracycline resistance, and TN5 is a transposon carrying kanamycin resistance. Now let's move on to deletions. Deletions are indicated by a delta symbol before the gene name, and if a deletion involves more than one gene, the delta goes in front of parentheses with the gene region inside. The whole region will include the genes listed and those in the segment that are deleted. An example would be delta, open parentheses, LAC, hyphen, pro-AB, close parentheses. This is the deletion of the lac-operon, genes pro-A and pro-B, and the genes contained in that region. Fusions are indicated by a phi symbol, a colon, or sometimes a dash with the gene name sometimes in parentheses. So it's a little tricky. An example would be GNTK fused to LAC-Z. This could be shown in parentheses with a phi symbol out in front or simply with a colon between the 2 gene names. Then there are inversions. A chromosomal inversion of a segment between 2 sites will be indicated with the capital letters I and V, and then the gene, hyphen gene in parentheses. So on the screen is an inversion of the sequence flanked by arrow A and FAD R. Prophage information is the next example of what different E. coli strains genotypes can tell us. A prophage or phage can infect bacterial cells, resulting in the phage genome integrating into the bacterial chromosome or becoming an extrachromosomal plasmid. Phi80, P1, and P2 are examples of an E. coli strain's genome that would indicate a prophage. Seeing Phi80 in the genome means the strain carries the lambdoid prophage Phi80. P1 indicates the strain carries the P1 prophage, and P2 indicates the strain carries the P2 prophage. And just for an example of a strain with a prophage is GB5 alpha. Here in its genotype, you'll see the Phi80 right up front. The next thing genotype can tell us about is restriction modification systems. These restriction modification systems can help provide protection from foreign DNA. The HSD-R and HSD-S genes encode an ECO-K1 restriction modification system that degrades DNA sequences that are not methylated at the adenine within the recognition site. The genotype also shows us information about other phenotypic traits such as antibiotic resistance and sensitivity. Antibiotic resistance is indicated by 2 to 4 letters for the antibiotic and a superscripted uppercase R for resistance. So AMP-R, for example, is ampicillin resistance, and CAN-R is kanamycin resistance. Sensitivity to cold and to temperature is indicated by a lowercase superscript, a CS for cold sensitivity, and a TS for temperature sensitivity. Cold sensitivity results in a loss of function phenotype only at lower temperatures, lower than a specific threshold temperature. Temperature sensitivity indicates exposure to high temperature results in inactivation. Lu A, superscript TS, for example, is the temperature-sensitive allele of Lu A. In the end, this is probably too much to memorize, especially from a video. The good news is that we have a great resource with this information for you. So if you're trying to recall something, you can always look at the article that we have, which will be linked below, and use that as a reference. I hope, though, that this kind of helped demystify strain genotype, and I hope to see you in the next video.